Dear colleagues, in my wrapping up presentation, I would like to tackle with a topic on what contribution could be made by the teardrops that we are using in the treatment of glaucoma to the neuroprotective effect. Well, first of all, we clearly understand, because until now we yet have no effective neuroprotectors both in CNS-based treatments and glaucoma. So this is I review treatment choices for newly diagnosed primary open angle and ocular hypertension patients. And there is just one paragraph here shown like this. So although there are lots of neuroprotectors shown in the experimental models, yet we have no option with evidence-based neuroprotection in the clinical practice. It is very important to remember, because to prove neuroprotective effect is extremely challenging and complicated. It's not like to prove hypotensive effect or ability to influence ocular hemodynamics. That is why in the clinical practice we have just single publications about that. So there is another challenge here. By now, there are no common standards, accepted standards of the assessment of the efficacy of neuroprotective treatment. And you know that until now, we use perimetry as the most important tool to assess progression of glaucoma. But in order to get um, evidence-based information, we need to have multiple tests. In this context, the technology designed by our British colleagues about 10 years ago, the technology of DARC, or imaging healthy and effective RGCs in glaucoma in the real time, where the dye is introduced here and the cells which are in the apoptosis already start flashing, is very perspective, because actually this technology really allows to assess the efficacy of neuroprotectors within a very short period of time, not years, but in fact weeks and months. And what is very important here is that this technology is at the phase 2 of the clinical trials, but it took actually 10 years to go through phase 1. So until now, we mainly face multiple talks about the neuroprotective properties of the drugs proved with animal models. Yes, here we see some progress. We see that there are special breeds or even special models where you can reproduce glaucoma process. So models shown here, and they're very well known. Moreover, today we know that pathogenesis of neurodegenerative glaucoma, and we spoke a lot about it today, is multifactorial, is very versatile. So influencing just one piece of this picture, of this puzzle, could be considered as direct antiprotector effect. And as the summing up of this webinar today, once again, I would like to tackle most important chains of pathogenesis in glaucoma optic neuropathy with the particular methods of treatment which are available, can be used and already used. As you understand, all speakers today emphasize the role of oxidative stress. In fact, this is a fundamental process which is closely related with the rest. So reactive oxygen species, they are formed in mitochondria during reperfusion because of dysregulation that Professor Wong was talking about. But exactly at that moment, we see the reactive oxygen species that could potentially launch the whole cascade of reaction of neurodegeneration. So here you can appreciate oxidative stress is in the center, cytotoxic phenomenon, autoimmunization, and glia activation that was covered in detail by Vera. So, and it all has mitochondrial dysfunction. That is why the search for treatments with antioxidant activity in the treatment of glaucoma may potentially be considered as one of the priorities in neuroprotection. In this context, there were quite a lot of studies devoted to the coenzyme Q10 as antioxidant protecting mitochondria. Also, it has been mentioned that coenzyme Q10 is much more effective if it is combined with other antioxidants, and specifically alpha-tocopherol, vitamin E. So here you see it is exactly what's proved with dark technology. So it means in the real time, we see the number of cells with apoptosis with the treatment of vitamin E in the center. And this is illustration B, and it is much higher versus the case where the cocktail from coenzyme Q10 and vitamin E is administered. It's important to understand that by now we already had fantastic drops, eye drops, that are effectively used by our Italian colleagues, and they already showed in the clinics that administration of such drops could improve the Q10 and PERG and the evoked visual potentials. In the experiment, we enjoyed Brimonidine antioxidant properties as it could activate alpha-2 receptors. We can create conditions to reduce the level of MDA and increased antioxidant protection. And this is, we see important antioxidants like cataplases and glutamate, but that was purely experimental work. So we also dealt a lot with experimental analysis with platelets and red blood cell model. We tested all drops here that could potentially be used in glaucoma treatment, and this is how I show you the 10-year data. So we found out that carboanhydrase inhibitors, Dervlovit, they have highest antioxidant activity versus others. For your information, you see different carboanhydrase inhibitors and their fixed combinations that are currently available in the national market. So please don't forget, and there are also some new options. And here, there is another review in two parts where I provided detailed data on carbohydrate inhibitors besides hypertensive properties, and the specificity of this group should be taken into consideration in glaucoma treatment. Where in the same models we checked fixed combinations, we also confirmed that the fixed combinations of the carboanhydrase inhibitors with timolol had highest antioxidant activity. And from the analogs of prostaglandins of fixed combinations, so the advantages were seen by latanoprost with timolol when we studied glaucoma. And actually it coincides with literature findings where separately we looked at the oxidative stress in the culture of ganglia cells, and stress was peroxide oxygen. So we studied the neuronal apoptosis. We saw that latanoprost and timolol could increase this neuronal survival in this oxidative stress, and especially in combination of both. The other classical phenomenon that all previous speakers mentioned about, so this is phenomenon of cytotoxicity, which is related with the increased penetration of calcium inside the cell, where there is excess of glutamate, so and this neurotransmitter, but there is too much of it, and Vera actually emphasized that Muller cell starts releasing glutamate as well. So finally, it leads to the excitation of the receptors, special receptors of, on the neuronal surface, and MDA receptors and gates become open for the internalization of calcium. And what does it mean? Specifically, these ions could launch the whole cascade of the irreversible death of cell related to the caspase activation. So caspases, these are enzymes, pro-apoptotic enzymes. So if we measure the level of calcium in neurons or the level of caspases, so we can judge how damaged is neuronal cell, and this is exactly what was carried out for many years, in fact, when we studied neuroprotective effect, including different options in glaucoma. So you clearly remember that one of the first neuroprotectors in glaucoma treatment was beta-xylol. And this is a very old study by Osborne, but in fact it demonstrated that with the effect of beta-xylol, we reduce the penetration of calcium inside ganglia cells. Some comparison of neuroprotective effect of adrenergic receptor drugs in retinal cell cultures was carried out, and it was shown that beta-xylol and brimonidine, but not timolol, provide direct neuroprotective effect, but not the timolol. So at that time, we couldn't have electronic microscopy or some other interesting studies that could be carried out now. So here you see glia, not on ganglia cells, but nevertheless, such conclusions were made, and for many, many years, we are saying that betaxolol, this is a direct neuronal protector. So there are multiple studies on the analogs of prostaglandins and mainly latanoprost. It was shown that latanoprost protects red retinal ganglion cells from apoptosis in vitro and in vivo. They looked at the penetration of calcium inside neurons. So with latanoprost, with culture of ganglia cells, we can reduce the calcium influx inside the cell and reduce activity of this pro-apoptotic enzyme caspase-3. In the experiment, it has been shown that with latanoprost in different models, so the mechanical stress exotomy or the model with NMDA, we see the increase of survival of neurons with latanoprost. And that was also the evidence of direct neuroprotective effect of the drug. So you see several publications that were describing these properties of latanoprost. In fact, there are even many more of them. We have to say that there are some analogs of prostaglandin. They were also studied, and specifically tafluprost. That's another prostaglandin analog, and the calcium penetration, calcium influx was assessed from the inside of ganglia cells. So, and it has been shown that with tafluprost, the effect is reduced. In fact, the working concentration was the same like in the vitreous body as standard molecule eye drops are used. Japanese investigators compared all prostaglandin analogs, and they showed that approximately all of them have very similar direct neuroprotective effect. And I'm emphasizing that was effect not related with the local hypotensive effect of prostaglandin analogs. So they discussed different possible modes of action, and among them the influence to the cyclooxygenase with the neuroprotective activity, and also the reduction level of caspase-3. And in fact, there are even more effects of this, and they continue to be studied. But honestly speaking, I have to emphasize that the maximum amount of the experimental studies about the anti-glaucoma drops was performed because of Brimonidine. This is a leader. So this is one of the first publication where after the clamping of the ophthalmic artery in the rodents, it was shown that Brimonidine could support the internal layers of the retina versus control. And that is exactly one of the first evidence with the direct effect of Brimonidine. In terms of cytotoxic phenomenon that we are discussing right now, it possibly may be related with the suppression of toxic effect of glutamate, with the inhibition of the apoptotic cascade and increasing of expression of anti-apoptotic genes, with the effect of alpha-2 adrenomimetic brimonidine. But the most interesting and intriguing studies were carried out with Memantine. And we know this is non-competitive blocker of NMDA2 receptors. So that's to a certain extent a dramatic story because the study is extremely expensive, recruited lots of patients on 4 continents that was carried out for 4 years and even reached phase 3 of the clinical trials. But it is exactly phase 3 that was negative, and there was no statistically significant difference of the direct neuroprotective effect of memantine for the glaucoma course, and the drug was not registered, was not approved by FDA, and until now it is actually not discussed as a standard of treatment for glaucoma. And the publication came to press only in 2018 when Robert Van Ray is analyzing not quite correct design of the study that did not allow to prove neuroprotective effect of memantine. Today, we spoke a lot about mitochondrial dysfunction and autophagy, which are closely related to the active oxygen species because mitochondria itself is the source of the active reactive oxygen species, but they also suffer from oxidative stress. We know now ways of the early identification of mitochondrial dysfunction using depolarization, GCI1, and this is a dye and which works, and this is the first stage of possible neurodegeneration. So different options are being studied, and I think the most impressive is the number of studies on acetylcholine, which is which was studied for the neurodegenerative pathology of the CNS, but also many studies were done also in glaucoma, and this is a literature, very recent literature review on this topic. And here you see multiple properties of this drug, and the leading one here is the recovery of mitochondrial function. Again, antioxidant and anti-apoptotic, suppression of glutamate, and in fact, they're analyzing the ability of acetylcholine to provide not only neural protection, but also neuroregeneration and neurorecovery. So in the clinic, there are multiple studies, I mean, ophthalmological studies, and I'm showing just one of them, where you can enjoy that acetylcholine inside the ROS could improve pattern ERK and evoked visual evoked potential. Here you see some other treatments that have capacity to reduce mitochondrial dysfunction. So all these drugs are very well known to you. Some of them are already discussed. For example, vitamin B3 is now very fashionable in the studies. And by the way, in China, they already initiated randomized clinical trial of vitamin B3. But again, curcumin, aspirin, resveratrol, green tea. The next stage that was covered by Vera is neuroinflammation. I'm not talking in detail about it, but this is a key mechanism of neurodegeneration, and the effect to this mechanism that we already spoke about in our discussion. We discussed complement, and this is possibly very perspective in the future. Resveratrol, as we may think. So it can be administered to our patients, possibly, because it has some inflammatory activity, so inhibiting anti-inflammation of microglia. But the problem is that it almost doesn't reach retina if you take it per hour, so just like curcumin, which is also well known in the South Asian medicine and cuisine for about 2,000 years, and actually more than 10,000 publications describes properties of curcumin. So to eliminate this mitochondrial dysfunction and to inhibit inflammation. But to what extent it could go to the neurons of the retina? So I'm happy to draw your attention to the very active design of new ways of drugs that would provide very high bioavailability. In this context, the most perspective here are the nanotechnologies. Nanocarriers are a special delivery system for the drugs, and materials could be very different. So missiles, lipids, polymers, polymer tubes, and classical examples are liposomes. So these are composite structures from phospholipids that somehow can put on other molecules on the surface and deliver them to the site, to the particular reaction site. So this is targeted delivery, and as a result, there is a dramatic reduction of the side effects, not only systemic but also local ones. And here, this is quite surprising that curcumin I just mentioned, already capsulated with nanoparticles, was already investigated in the clinical practice, and this is phase 2 data. And these are the drugs, this is the bioavailability which was studied, but not in glaucoma, not in ophthalmology, but in Alzheimer's disease, and with very prospective results. And here you can appreciate curcumin, which was studied in glaucoma, but only in the experiment. And here again, we are supported by DARC technology. Using this technology in the online regimen, we managed to show that in a single droplets of this treatment, as nanoparticles of curcumin manage to reduce the death rate of retinal ganglia cells, and these data already published. Memantine that we just discussed, appears to be already well-designed form of the local effect of memantine, again with nanoparticles and with pretty high bioavailability. And I'm emphasizing bioavailability of memantine to the ganglia cell. And again, using DARC technologies, it has been shown that the teardrops with these nanoparticles already within 3 weeks can improve the survival of ganglia cell of the retina. This is experimental publications. So very perspective are soft contact lenses as a delivery system for the drops. Because inclusion of the colloid nanoparticles, I emphasize nanoparticles into the polymetric matrix of the contact lenses could provide well-regulated and long-term release of the product. And this is important from the point of prolonged neuroprotective effect. Speaking about contact lenses, let me say a few words about the surface. So I remind you, so all the drops that our patients taking actually damaging the eye surface. So we need to think about the protection of that. And for example, Stilavit was studied with confocal microscopy. It has been shown that within 3 months, we see the recovery of endothelium, and this myth creates special protective film due to the high elasticity during the blinking, which is important area for the protection of the corneal surface. We know very well that many drugs that we administer for the local hypertensive treatment may lead to the dramatic adverse events that forces patients to refuse from the treatment. That is why currently there is active search of new delivery system for the treatments. And for example, for bimatoprost, you see special implants offered that could be actually delivered into the ciliary groove, and then the drug is released within 1 year. So then biodegradation happens. So within this time, there is a reduction of uh, intraocular pressure. This is Allergan design. So these are available drugs with the matoprost fixed combinations plus preservance free. And please appreciate this is Bimicombi anti-glow echo. So this is not just the comfort. So this is a very effective solution. Travoprost, so that's another prostaglandin analog that also unfortunately has local adverse events. That is why the implants were designed for the microscopic delivery system of the treatment. Unfortunately, illustration is not completely shown here, but it reminds stent implantation, which is microinvasive glaucoma surgery, where direct to trabecular there is microstent implanted. So here, it is a drug which is slowly released within next 6 months. And again, these are available options with travoprost on the market. So there are other delivery ways for the drugs. Here you see we can even introduce them on the conjunctiva. So then they could work for about 3-4 months. If you implant on the conjunctiva, it could be 6 months or 1 year, or it could be eye occluders. So these are all different delivery ways that can also be available for the potential neuroprotectors. And last but not least, neurotrophic factors that we spoke a lot today about. So extremely interesting presentation by Alexander, by other colleagues. You understand how important they are in the pathogenesis of glaucoma and in the treatment of glaucoma. I would like to add that these multiple neurotrophic factors are effective only in a case if on the surface of the neurons of the ganglia cells there are receptors which are sensitive to them. So these are first of all tyrosine kinase receptors. And if cell is depleted and losing its uh, receptors, so there would be no effect of neurotrophic factors. So this is very important study, experimental study with Brimonidine shown here. You can appreciate that that 2 days after intravitreal introduction of the product, we see threefold increase of receptors sensitive to BDNF on the surface of ganglia, retinal ganglia cells. And this is brain-derived neurotraffic factor. Also, another study for Brimonidine, and this is a model with the experimental ophthalmic hypertension, which actually proves that due to Brimonidine, there is activation of receptors of ganglia retinal cells sensitive to different neurotrophic factors. And here we even discussed the combination with axons. A very important conclusion was made from the experimental studies with Brimonidine, and I continuously wanted to draw your attention, and I was asking questions to our speakers when to start this treatment. So definitely the treatment should be started as soon as possible before we see irreversible neuronal death. And this is exactly what multiple studies prove and multiple reviews on Brimonidine. Here you can appreciate products available with Brimonidine. Unfortunately, we know that there are local adverse events which are quite frequent for alpha-gun and combi-gun for alpha-2 adrenomimetics. But here I would like to draw your attention that currently there are drugs available with preservance-free, and Brim anti-Glaucoma is one of the new options, and Brimine Zedua. So these are direct neuroprotectants. Today, Alexander already was saying that there is a special implant that could release growth factors, and this is ciliary neurotrophic factor. This implant is located in the vitreous body through the flat part of the ciliary body. It is fixed, then you can remove it. But the point here is that neurotrophic factors are slowly released from the implant. And the ideal idea for the neurotech company is to implant not simply the neurotrophic factors, but let's say simultaneously to the receptors which are sensitive to it. And then the release of this cocktail would happen when there are some impulses to ganglia cell about apoptosis, where this neurotrophic effect is highly required. And in 2021, the phase 1 of the trial has been completed, so yet it is not in the practice. Nasal spray delivery system. Mikhail already mentioned it today, and this is also very interesting design. So there are multiple vessels in the nasal mucosa, and with the example of the treatment that was obtained from amniotic membrane. So, and then they will be able to identify neurotrophic factors in the nasal delivery. And this is not just possibly going into the eye, but possibly into CNS. And this is another quite interesting delivery way. Cell technologies. Well, we had great expectations, but unfortunately now it becomes more than obvious. Neither Muller cell nor polypotent cells can be transformed into the ganglia cells, and even mesenchymal cells did not show this effect, so they cannot be transformed into the retinal ganglia cells, and this has already been mentioned before. But this is more than obvious that these cells, they are capable to release the same neurotrophic factors and improve neuronal survival by 30%, which is very important. And quite obvious question, and what about clinical practice? I already spoke about the studies of Memantine. Then, for many years, the active studies of neuroprotectors were actually stopped. And there was such a review by Barrick quickly that clinical trials for glaucoma neuroprotection are not impossible. So he just explained how many spirometries should be made in order to assess neuroprotective effect of the treatments. By now, it has already been implemented in a number of styles, specifically in a UKGTS, famous British study with miltenopros, showed that it can, versus placebo, reduce progression of glaucoma during the second year of treatment. So here you see the statistically significant difference. So this is real-world data with neuroprotective effect of the treatment. More advanced design was shown in a very famous study with Brimonidine. This is um, low GTS study, and that was comparison with Timolol head to head with a very similar antihypertensive effect, but the progression was one-third lower versus with Brimonidine. And last but not least, very fresh data, freshly published in the journal Glaucoma journal in July 2020, which is devoted to ceticolin. Ceticolin was studied in clinic peros, but here it's eye drop with ceticolin. The question whether the treatment with ceticolin could reduce progression of glaucoma, and that was multi-center randomized placebo-controlled study. Placebo is important. So it has been shown that with ceticolin, we saw dramatic reduction of glaucoma progression rate, which was assessed by the light sensitivity of the retina shown here, by the progression rate for MD index, and also reduced rate of the retinal ganglia cell thinning. So this is how this trial demonstrated direct neuroprotective effect of ceticolin in the clinical manifestation. So, colleagues, wrapping up, I would like to say the following. T-drops that we are using locally for the local antihypertensive glaucoma treatment provides direct neuroprotective effect, although clinically it has not yet been fully proved. Neuroprotective treatments should be started as early as possible. And first of all, it should be recommended to the patients with a fast glaucoma progression. For that, you need good quality monitoring. And it is very preferable to administer the drugs with multifactorial effects. That means to provide influence to different chains of this multifactorial pathogenetic influences. And definitely there are great expectation towards new pathways, new modes of delivery, and design of new teardrops based on neuroprotectors. But yet, this is studies for the future. With that, thank you very much for your attention.